of Nightingale, both great colleagues whom we have worked with now for a while as members of the IHCC. And they are going to present to us today on the Nightingale Biomarker platform on the profiling sort of across large biobanks. UK Biobank obviously has typed enormous number of samples. We have done this at the IHCC in a limited way, but we have actually really compelling data, we believe, from the data sets we have used. So the sort of rest of the title here is From Molecular Insight to Advancing Preventive Health Screening. That's exactly a topic that we have keen interest in, and I'll just welcome you both. Thank you for taking the time to present today, and we look forward to your talk, and then we will have a discussion afterwards. Please go ahead.
Yeah, thank you very much. Thank you for the invitation and the opportunity to speak here today. We're presenting some exciting new science coming out from these large biobank initiatives that we have ongoing. So my name is Peter Würz. I'm not the CEO, but I'm scientific director and one of the co-founders of Nightingale, and I've worked with many of the research collaborators over the years in many cohort studies. Initially, we started out mostly focusing on cardiovascular disease and diabetes, but we are very pleased to have really expansions out to many, many different types of diseases that we'll show you some examples of here today. And moving on, I think, from the kind of molecular discovery into really pushing the frontiers to a precision medicine. And I think we share a common goal on that. And my co-pilot here today is Dr. Jeffrey Barrett. He will run the second part of the talk on this predictive analysis, but I'll start us off. So Nightingale Health has developed this blood biomarker testing platform that has really been widely used in cohort studies, initially mainly in Europe. And we've listed some of the flagship projects here: UK Biobank, Estonian Biobank, Finnish cohort studies, and Netherlands, and so on. But very excitingly, we see now this is being more and more used across the world in studies from Latin America, and in particular in Asia, there's been a lot of interest. So we hope to really kind of have, in the same way as in genomics, sort of join forces with these biobanks and advance the science in this way. In particular, I'd like to highlight the IHCC project, which, as Håkon alluded to, we've done a successful pilot with exciting findings and seemingly good data quality, ranging from pediatric studies to cardiovascular focused cohorts from Iran and Bangladesh. And we're pleased to have this extending also to studies from Africa and South America, where the platform hasn't almost at all been used previously. So really exciting times. And in the end, we can sort of discuss how this can be expanded further in IHCC. But for now, we'll first give an overview of the biomarker profiling platform and show some of the scientific highlights now coming out from the UK Biobank project. That is arguably our flagship project. Really, really exciting times. We are profiling the whole cohort and getting good use of the data, getting out to the community. So, but before we go into all the science, just sort of like step back a step or two and sort of think about why are we doing all of this. So the mission and vision of Nightingale is to do population scale biomarker profiling to advance preventative healthcare. And so in order to understand the relevance of all of these biomarkers for the individuals, we first have to profile the biomarkers in very large cohort studies and biobanks. And that's where we really have common goals with you all. And we really see this now, the data is really delivering in terms of evidence for improving early disease detection. And how do we kind of get to profile these very, very large cohorts? So we've kind of developed this biomarker profiling platform with the aim to measure basically full cohorts in even the largest studies in the world. So we focused on a platform that can deliver on high throughput with only small blood volume needed and really optimizing for cost effectiveness. So the high throughput is enabled by a platform using a nuclear magnetic resonance spectroscopy, really optimized to rapidly screen through thousands of blood samples. And then small volume, so only 100 microliter is needed, either serum or plasma. And then we try to optimize the protocol, so the price goes down to around 20 euros per sample for large studies like IHCC cohorts. And kind of pleased to hear the feedback from kind of the initial results on this, and then kind of we can really extend this to very much larger scale across IHCC. So what do we measure? The Nightingale Biomarker platform with NMI delivers a broad metabolic profiling platform. There are many lipid measures, including routine lipids and more detailed lipid measures, fatty acids and amino acids. And I particularly highlight the 39 clinically validated biomarkers that we deliver that are approved also for diagnostic use and return to participants. And in addition, there are many more measures that are relevant for research use. But rather than going into the individual biomarker names, I think it's helpful to think about the platform and what it delivers in terms of molecular insights or sort of overall metabolic pathways that it captures. And here I've highlighted some of the main aspects on the terms of the detailed lipid metabolism. The insulin sensitivity is captured quite well, reflections of dietary intake and chronic inflammation very strongly. And some recent data also shows that some of the biomarkers relate to immunity and fluid balance, like kidney function and other measures of fluid balance, and then metabolic processes like glycolysis, ketolysis, and even mitochondrial function. And that together means that these biomarkers is really relevant for disease risk. We know all that these pathways are relevant for many diseases, and in particular, it becomes even more relevant. You combine information from multiple pathways in one go, and as Jeff will detail later on, when you combine the biomarkers together, you can even get better prediction than you get from any individual biomarker. One aspect on the individual biomarkers I would like to highlight, however, is this aspect that the Nightingale panel basically includes clinical chemistry measures. So we have calibrated the cholesterol measures, apolipoproteins, creatinine, and glucose measures to really match with the same levels as you get from clinical chemistry measures. So in your large cohort settings, you can basically completely replace the need for the standard lipid testing and some of the other routine clinical biomarkers if you run with the Nightingale platform, because these measures are included in the platform along with the more emerging biomarkers. And so on the data here, we've had some results from the FinRISK cohort studies where you see very good match both in terms of correlation, but also the absolute levels with clinical chemistry. So these results I show you again here on the left-hand side is the results from the FinRISK study where we compare the ApoB measures with clinical chemistry compared to Nightingale on the x-axis. So you see a really excellent match here. However, I would like to point your attention to the fact that if you compare these measures in the UK Biobank, here shown on the right-hand side, you see slight differences in the absolute concentration. So the correlations are still very good, more than 0.9 correlation, but the clinical chemistry tends to be slightly higher absolute concentration than what we measure from Nightingale. And that is because a dilution issue happened with the UK Biobank samples upon the sample collection. And that means that the different aliquots within UK Biobank are diluted to different degrees. And the aliquots that were measured with clinical chemistry are different from the ones measured with the Nightingale NMR platform. And that is the main reason for this kind of more scatter than you see here. However, for epidemiological use, the association testing of the biomarkers in UK Biobank is really consistent whether it's NMR, Nightingale data, or clinical chemistry. So here we show some results on future risk for osteoporosis, and you see that the association magnitudes for the different overlapping measures are essentially identical, whether it's clinical chemistry or Nightingale NMR measures. And that actually is the case even for a measure like albumin here, where the correlation in UK Biobank is quite suboptimal, I would say, is less than 0.6, but still you see that both of them are fairly good predictors of future risk for disease. So it's sometimes good to think about not only what is the correlation with other biomarker panels, but how well do they match in terms of predicting future disease risk. All right. So moving on from the technological aspects into some of the new science coming out of UK Biobank, there's lots of papers now coming up almost every week. There are new preprints being posted when the data on the first 100,000 individuals has been in the public domain for a bit more than a year by now. And we see many applications of studying of, for instance, causal inference, combinations with genetic data, characterizing known and novel drug targets with genetic data, and really many exciting different applications that we never dreamt of would be possible, and certainly within Nightingale Science team do not have bandwidth to analyze. But perhaps most excitingly is these new results on the disease prediction that we'll come to in a second. So I'd just like to point your attention to this really nice paper in Nature Medicine on multidisease prediction that we'll come to, but much more, many more opportunities in that space. The main paper I want to focus on today here is, for my part, is this atlas of biomarker associations that we put out on medRxiv in the summer and was just accepted last week in Nature Communications. So here we looked at the biomarker associations for all the 249 biomarkers in the panel against more than 700 different disease outcomes with, I think, more than 50 incident disease events. So here we can really characterize for each biomarker what is the sort of disease specificity or even lack of disease specificity for kind of across the span of different diseases characterized by the different ICD-10 codes. And we can also, if you are like more interested in a specific disease, say, I don't know, fibrosis or various eye diseases or whatever, cancer or something of your particular interest, you can look up what is the overall biomarker association profile of that disease. So we encourage you to go and check out this web tool and to sort of explore all these exciting findings. I'll just highlight just very few of them. So I think the main take home from this analysis was that how widely the biomarkers associated beyond diseases of the circulatory system and metabolic diseases like diabetes. So this figure shows you for each of the 39 clinically validated biomarkers, how many significant associations there were across the different ICD-10 chapters. And you see on the top here for some of the fatty acid measures, associations with infectious diseases, various cancers, many circulatory diseases indeed, but actually as many diseases of the digestive system. So really, while we initially had this platform widely used in heart disease research and diabetes, I think there are many applications that are now opening, and we're just kind of scratching the surface in terms of understanding the biology of this. Some of the other biomarkers like amino acids were much less kind of associated across the board of diseases. So it's not there's kind of quite a bit of differences between the individual biomarkers. But sort of what we see is kind of some of these like inflammatory biomarkers or fatty acids, they really are associated with many different types of diseases, diabetes very strongly, but also different diseases like gout, smoking, alcohol-related disorders, sleep, kidney function, hypertension, pneumonia, osteoarthritis, and so on. Really diverse span of associations. And some of the fatty acid diseases are as broadly associated with many diseases as the inflammatory markers. Another important finding is that the not only looking at the statistical significance, but in terms of the disease association magnitude, how strongly many of these biomarkers are associated with this sort of non-cardiovascular type of the diseases like risk for sepsis, lung cancer, and depression was all often as strongly associated as future risk for myocardial infarction. Another insight from this kind of figure comparing the different overall biomarker signatures for the different diseases is that many of them actually have quite similar disease association profile that kind of may also be relevant in terms of prediction analysis. So overall, very, very exciting findings from this biomarker disease atlas. We hope with this tool that we can really kind of take some steps further in biomarker research and saying, okay, it's not so much about the biomarker discovery, but with this data out in the public domain, you can better zoom in on the molecular function, what is the kind of etiology, and really move on towards risk prediction or combinations with different studies of different study designs and across different ancestry to really advance this exciting science. So enough for me. I'll hand over to Jeff to talk about some of the prediction aspects.
Thanks, Peter, and thanks, Håkon and IHCC for the invitation to present today. So yes, I'll take over and start talking about how we are building on this huge resource of biobank data that's deeply profiled to think about prediction and how that can be translated both in research and we hope in the near future into precision medicine and real clinical use cases. So as Peter said, a kind of core insight that came from looking at the UK Biobank data is that many of these key metabolic functions that we profile are involved in many different diseases, and it's really about combining these in a data-driven way to try to understand how they can be best used for prediction. So I'm going to start with an example that was published last year in work that was led by Peter and a very talented colleague here in the company, Heli Julkunen, and it's looking at severe infectious disease. So it was obviously a really important focus area in the last couple of years during the pandemic, and was the first example, I think, that Nightingale showed moving outside of that cardiometabolic area and into showing this is really relevant in surprising places. So the data that we work with now, the published version is based on that 100,000 sample data set that's already in the public domain. We internally now have increased that to 300,000, which is what I'll show some analyses on later in this talk. And that 300,000 sample data set will be available to the public in the middle of next year. So these are all samples who gave blood to the biobank approximately 10 years ago. So the way this study works is you have a snapshot of their metabolic health from when they were enrolled in the study, and then you look at the electronic health record follow-up data in the subsequent 10-year period. So it's really a prospective look at, can we build models using the blood sample taken at enrollment to predict the risk of disease in the future? And some of the people that, as Peter alluded to, there's lots of outcomes we can study, but some of them, of course, were hospitalized with a severe pneumonia. For most of that 10 years, the cause of such a pneumonia would likely be influenza or perhaps a bacterium or something. Obviously, in the very recent years, there were a very large number of these caused by SARS-CoV-2, COVID-19 disease. And essentially what we do is split the data set in half. We build a machine learning model to create a multi-biomarker score that predicts the outcome of interest, in this case, severe pneumonia. And then we look to see how well it does in the independent remaining half of the data. It's one of the beauties of doing this in such a huge cohort that you really have extremely good sample size to both train the model and then evaluate it. And if you look in the case of severe pneumonia, much like some of those forest plots that Peter was showing, many of the biomarkers are associated with future risk of being hospitalized with pneumonia. But really the key thing is that once you create this multi-biomarker score, it is more than twice as predictive as a future risk as any individual biomarker. So really highlighting this observation, which turns out to be quite general, that it's in these kinds of biomarkers, at least, it isn't really the case that you find the one true perfect thing that predicts it. It's really a combination of multiple sort of dysregulated bits of metabolism that are picked up by different measurements in the panel. And this is, my background is for a very long time in genetics, and it's, I think, very analogous to what we've discovered in essentially all chronic disease, that the genetic risk is spread across many different pathways, and any individual patient doesn't really have like a single genetic problem, but some amount of perturbation in multiple pathways. And one way to look at the performance of this is, so you, as I said, train the model in half the data and then evaluate in the other half. So each of these dots is 1% of the test data set. So in our current release, each of these dots is 1,500 people, and you rank those percentiles from the predicted lowest future risk based on the biomarker score on the left to the predicted highest future risk based on the biomarkers on the right. And then the y-axis is over the 10-year follow-up period, what proportion ended up in the hospital with pneumonia. And you can see that there's a kind of linear relationship throughout, but really the dramatic action is happening in the sort of 80 to 100th percentile, where there's just this tail of people who are at substantially elevated risk compared to the average person in the population. And one way to look at it is if you look at the sort of top 10% of this tail, they're at 5-fold increased risk over a 10-year period of being hospitalized with pneumonia. So that's really a pretty substantial risk factor. It's one that we don't think is obviously captured by, sort of other things you might look at in a general population. And it starts to give a hint, I think, that if you had this information available at like a population scale, you might be able to, alongside with kind of population primary prevention we're more familiar with, like, for example, finding people at increased risk of cardiovascular disease and getting them on statins, that for people in this high risk tail, you might want to make sure that they get their influenza vaccine every year, that they get their COVID booster every 6 months, these kinds of preventative things in the same situation. And indeed, in this paper, if you look at the data's trained, because it's historical data, there are more cases of non-COVID pneumonia. But then if you use the same score to predict the cases of COVID that happened in 2020, which is like on average almost 10 years after the blood sample was taken, the strength of the association is almost as strong. These x-axes are not exactly the same, so it's about 1.4 per SD in COVID versus 1.7 or something for other pneumonia. But most of that, I think, is about the time difference in the outcomes. And just to emphasize again, this isn't about risk of getting infected with any particular infectious agent. It's about, if you do, how sick do you get? And that's really the thing that you can try to stop. And I think this does mean that, again, outside of the existing well-known primary prevention cases, there are opportunities to do this in other circumstances, ones that might not have been so obvious a priori. And what we can now do is essentially do this in many, many different diseases, which have a good multi-biomarker score. So this is the same kind of look of a 1% per dot plot, ranking how high the risk is, or sorry, what is the actual event rate as ranked by your risk in the score. And for this wide range of outcomes, there is really a quite substantial amount of information in these scores. If you measured one blood sample, you can, of course, produce all of these scores from the same data. So it means that you can bring the idea of primary prevention to a wider range of outcomes than I think is currently thought about in healthcare systems, without doing a big, complicated set of additional tests. Because we focused, as Peter alluded to before, on getting the price down, you can imagine replacing a kind of standard lipid test in an existing population screening program. Like in the UK, they do one for people who are over 40 for cardiovascular risk, for not much actually marginal increase in cost compared to just doing the lipid panel today. And then you could generate these other risk scores across a wide range of other outcomes. As I mentioned, I'm a geneticist, and I started thinking about genetics in this immediately when I moved to Nightingale a little more than a year ago. And there's a very strong analogy between these biomarker risk scores and polygenic risk scores, which of course have a lot more kind of basis in research and lots of interest in investigating. And so one slightly different way of looking at the same data from the previous slide, and I should just note that these 8 diseases that are shown here are not chosen at random. These are essentially the top 8 sources of disability adjusted life years in Europe, according to the WHO, other than a couple of sort of like non-chronic accidental things like falls and back pain, for example. So we're trying to focus on a set of diseases which are the really important ones from a public health, or at least a set of important ones from a public health perspective. And the fact that we have some information in so many of them, I think, underlines the potential here. But so these are looking at basically the gray line is over the 10-year follow-up, the rate of these different diseases happening in the population. The blue line is the top 10%, that kind of tail that we discussed in the previous slides of risk in each of these by the biomarkers. And the red line is the top 10% by a polygenic risk score based on the kind of current best published result. So you can see that the biomarkers provide a lot of information that is complementary to the polygenic scores. And it kind of makes sense, I think, because the genetics is capturing some fraction that we currently understand of your inherited risk, and the blood biomarkers are capturing a combination of inherited predisposition and the kind of integral of your entire lifestyle of up to that point and how that has affected your risk. And another thing that I think is interesting from these is that even though there is more information about your risk of, say, developing diabetes in the 1, 2, or 3 years after you give the blood sample, there continues to be substantial information in years 7, 8, 9, 10 after the blood sample. In other words, the lines on this graph continue to diverge 10 years into the future. And that, I think, is interesting because it means that this snapshot of molecular health now is really predictive of what's going to happen to you far into the future, which is not, I think, typically how in a clinical setting some of these markers like cholesterol levels are used, where it's like, I'm looking at this value now, maybe I want to act on it. If I want to think about it again in the future, I'll measure it again. But actually, each measurement does carry some information about your future risk. And I kind of alluded to this before, but you can imagine this QRISK is the version of a risk calculator for cardiovascular disease that's used in the UK National Health Service screening program. It incorporates family history, smoking, diabetes, BMI, age, and only cholesterol as a molecular measurement. And if you dropped in the Nightingale test, you can see that it's already quite good. The top decile in QRISK does identify people who have a much higher event rate than the full population. If you drop Nightingale in to essentially replace just the molecular component, we improve that even a bit more. One way to think about this is not just that you're finding people who are at even very high risk, because already you'd want to clearly intervene with, say, statins or maybe blood pressure lowering medication to those people, but it actually reclassifies a non-trivial set of individuals where it finds people who under the current paradigm would be classified as sufficiently low risk not to intervene today into being high risk, and that group that the Nightingale boosted paradigm has an event rate that's equivalent to the high risk group that is already prioritized. So this is sort of a way to maybe get into a situation where these kinds of ideas are already used, and we could drop in a way that's quite analogous to how they're currently used, laying the groundwork for the kinds of things which I think are really exciting about then being able to offer these multi-biomarker risks for a wide range of outcomes. So let me just wrap up. I want to just note there was a really nice paper from a group led out of Berlin that was independent of Nightingale, but used the data of the 100,000 participants in Biobank, and essentially took exactly the same approach that I've just described about modeling the risk of future disease. And as someone who was in academia for a long time, it was at first very depressing to be scooped by such a very nice paper before we had published our version of the prediction paper. But actually, it's also very useful for us to see other people in the community kind of seeing the same opportunity essentially that we do. And in fact, they had a nice note in the discussion that highlights, I think, the same thing that I've been talking about for the last few minutes, which is that this NMR metabolomics, because it measures a diversity of metabolic function relatively cheaply, has this opportunity as a single multi-disease assay that can find lots of people at future risk that are currently not clinically diagnosable, and ideally implement those in a situation where you can bring either lifestyle or medical interventions into play much earlier and more equitably and universally than we currently manage to do. So we've talked about some of the things that we are most excited about these data, but I think really this momentum of now being able to run at scale in these big biobanks opens a ton of different kinds of studies, some of which other investigators might follow up in the biobank data, but others are really not possible in the UK Biobank. For example, looking at diversity across the world. We have some early results that we're hoping to preprint maybe in January on the portability to different ethnicities of these biomarker scores and how that compares to genetic scores. There's also, I think, a lot of excitement from the things we were just talking about involving, using these to get individuals to change their future behavior by feeding back their risk information. UK Biobank can't do that because it's not part of the consent, but many cohorts do have that kind of opportunity. And I think that's super exciting as well. So thanks very much again for your attention. We think as well, there's great opportunities potentially with IHCC. We'd love to support your science. We think there's a potentially very good fit between the model we've built and how IHCC is working. And please do feel free to obviously have some discussions now, but also get in touch with us afterwards if you'd like. Thanks very much.
Yeah, great. Thank you, Peter and Jeff. I mean, this is actually fascinating, and I had sort of not really paid much attention to the biomarker score before, Jeff. And isn't it true that not only can you generate risk score, because many of these biomarkers, lipid biomarkers, are protective? Couldn't you also drive a protective score that sort of could be equally as informative in terms of informing interventions as the risk score? So maybe also in terms of the polygenic risk score versus the biomarker score, do you think that they are mutually exclusive, synergistic, or is the polygenic risk sort of capturing a part of the biomarker score?
Those are excellent questions. The first one, I think, is a really cool idea, which we have not looked at, or at least not in my time here, have looked at it. I don't know if you have before, Peter, but one thing is many of those risk curves have this hockey stick shape where you have some subset of individuals who are at really high risk, and then everybody else is at some sort of similar low level of risk. We have not yet seen an example where you have like a S shape, where you could really find the low risk people as well. But that would be very cool, obviously, if you could, because you'd want to know what's different about those people.
Right.
Do you have anything you want to add to that?
Yeah, I think exploring that in sort of closer to clinical settings here, we see the disease event rates in the healthy population cohorts is quite low. But if you look at sort of trial populations or kind of sort of I don't know, say pre-diabetes or whatever, kind of just before an event, then you may see these things more.
And then in terms of the complementarity, sorry, I meant to say that. So thank you for reminding me to say it, which is they are very complementary. When you add the two scores together, you get a further boost in the predictive accuracy. It varies a little bit by the phenotype. So in diabetes, for example, the genetics and metabolomics are a bit more correlated, which kind of makes sense because we measure glucose as one of our biomarkers, and some fraction of the diabetes genetics is purely about aspects of glucose synthesis and processing. And so there's some correlation, but in other cases, they're really very weakly correlated. And in all cases, you definitely get synergy of adding the things together. One thing which we're kind of interested in exploring now is many of the biomarker scores are moderately correlated with each other because these different pathways are kind of involved in multiple diseases. Right. And there's an interesting question of, if you have an individual who is sort of metabolically unhealthy, does their genetic profile help you stratify what's their kind of highest, most likely outcome that you want to try to prevent?
Yeah, no, I mean, that's great. I totally agree with that. I see that there is a question in the chat directed at you, Jeff, from Albert. So does how people rank by risk change between the QRISK-2 and the QRISK plus Nightingale?
Yeah, so I did a bad job of explaining this, but that was part of what I was trying to say, which is the operational use of QRISK-2 is you fill in the risk calculator, and basically if you're above a 10% 10-year risk of a cardiovascular event, the doctor will try to do something. And it might be a lifestyle intervention, or it might be statins, or maybe something else. And so that, one, I think, meaningful statistic is the sort of reclassification about people who are basically over or under that bar. And of all of the you capture like a substantial number of the future events in that high risk group, but it's not all of them. And if you look at the people who, by adding Nightingale, get reordered from below the kind of risk bar to above it, you get almost as many events again as you did by the original QRISK implementation. So it really actually has a potentially pretty substantial impact on the operational outcome of running such an improvement. Of course, when you do that reordering, you also, by adding more information, you kind of push more total people above the high risk bar. So you're also treating a slightly larger fraction of the population. I don't have the numbers in front of me, but when you look at it, I think it's pretty compelling that the additional true positives are worth the fairly small amount of what at least appear to be false positives, in the sense that in the 10-year follow-up, they don't have a cardiovascular event. But actually, I would predict that those people are pretty sick, and putting them on statins isn't a bad idea, even if they didn't in this particular study end up having a heart attack during the follow-up.
Yeah, that's great. Now we have an army of people on the call who can all unmute and ask questions. Are there any questions from the audience for either Jeff or Peter?
I have a question for Jeff and Peter, and that's around, I think you mentioned this, that you've started to look at ancestry or ethnic dependent effects on these risk profiles. Are you seeing very large effects, or is there sort of maybe an amelioration when you're looking at a metabolome versus a genome, where the diversity in the genome is much greater than the diversity in the metabolome, because you're only looking at 249 things?
Yeah. So for some things, they are kind of exquisitely matched across. So sorry, just to say, what do we have to do an analysis? So in the UK Biobank, about 10%, I think, are non-European, something like that. Yeah. Maybe a bit more, 12 or 15%. So in a 300,000 data set, that is some tens of thousands. So it's enough for the common outcomes to actually have a look. It's the first time we've sort of been powered to do it in the UK Biobank data set. So for some of the things like cardiovascular disease, the metabolome prediction is like identical in the different ethnicities. And maybe that's not surprising, because like that one, we have some nailed on causal risk factors like LDL in the predictor, so you sort of would expect biology to sort of work predictably. Diabetes is an interesting one, which is we're still investigating, but seems slightly less consistent. The prediction is a bit less good in the non-European subset. Still very strong, still better than the genetic predictor, for example, which is, has this complication, a much bigger complication of not being translatable from one population to the next as a rule. One thing which, if there are people on this call who are more expert than me, as we've looked a little bit, there are interesting, examples in the literature where like other risk factors, classical risk factors, sort of seem to behave differently. BMI, for example, does not have the same effect size in the South Asian population as it does in the European population. And so that could be part of what's happening, because our biomarkers are, partially picking up signals of obesity and stuff like that. So I think it's a bit open. I don't know, Peter, if you want to elaborate.
Yeah, I think there are many ways to look at this. Kind of one thing is to study are the kind of biomarker associations different in the different ethnicities, but one can also sort of look at it different ways, like are even like the concentration levels different, which may be more like due to environmental factors. And that is somewhat challenging to study just kind of across different cohorts with slightly different collection protocols. But now with the many different designs in IHCC, I think there's kind of new possibilities to do that.
Yeah, I totally agree. And certainly, I mean, it's fascinating sort of, to look at the cohorts we have done, and now we have a couple of African cohorts that sort of will be run shortly, and because the levels we see, for example, the cohort we did from CHOP, I mean, they were all African Americans, and so when we compare sort of the values in these individuals with reference values from the UK Biobank or other published data or other data from that were done in the IHCC data, there's actually quite a bit of difference. There's quite a bit of difference. And then when we dissect that down into disease level of sickle cell disease or asthma or all of the conditions, I mean, there are totally novel biomarkers that come up that have never been associated with these diseases, but they are, clearly involved with vascular biology. And, certainly in sickle cell disease, you have all kinds of, vascular events in these individuals. They get strokes, they get, acute chest syndrome, which, is with vasculitis and all the things. So just that little data set is actually very, very informative, from a biology standpoint. And I think we'll learn a lot more as we grow the numbers going forward.
Yeah, indeed. Then there's a good question from TM asking what exactly Nightingale help for someone with a cohort of size around 5,000 samples. So I think kind of one can view this in multiple ways, kind of as we alluded to in the beginning. You get standard biomarkers like cholesterol and apolipoproteins, creatine and glucose at broadly similar price as the standard clinical chemistry. In addition, you get all these other more emerging biomarkers that can do both kind of like new molecular insights and kind of be, then in a way you get a sort of a ticket to be part of the sort of omics revolution and sort of first mover in precision medicine. And I think, you will probably still measure standard risk factors or something like CRP or blood pressure or a GWAS panel in your cohort of 5,000. So just because this is now being done at big scale in UK Biobank doesn't take the value away by doing it in the smaller studies. Rather, I think that each study often have a unique design and can add some kind of contributions to the science. And then when you combine this Nightingale data with other data, say dietary measurements or subclinical measurements of disease, you can do a lot of new science without having the sort of biomarker versus disease as the main focus, but really uses as a tool to just better inform your other scientific studies.
Yeah, and I think, what we hope to provide is a kind of, Rosetta Stone of the same data that will, because it's relatively inexpensive and high throughput to produce, that will allow all of these studies to kind of interoperate and interconnect. And, Håkon will remember from now probably, I don't know, it was 15 or 20 years ago in the kind of early days of trying to do GWAS combinations, people had slightly different chips. You did some replication, targeted genotyping. Combining these things was a pain in the neck. And then, as it became possible to say, okay, the company's got the GWAS array prices down to the point where you could get everybody to be doing this and imputing and being on the same backbone, then everything became much more smooth. And I think for metabolomics, this offers the kind of same opportunity. Whereas obviously you can't, there are all these other, you could do a 2,000 protein panel, but if it costs you $1,000 a sample, then it's, it's going to be very difficult to get that sort of across many cohorts and in the entire cohorts.
Right. I know I did refer actually a customer to you not that long ago, but it turned out that he had rat samples. And I see one of the questions in here is, what about other species, cattle, pigs, dogs? Yeah. Is that on the horizon or?
So Nightingale has really focused on the sort of a technology for high throughput profiling, a sort of like the full cohort solution. So indeed, if you have 5,000 samples, you can you basically forget about doing a pilot case control study, just run all 5,000. It's totally possible. But for that reason, we've also haven't really focused on sort of the animal studies that are often much smaller in sample size.
Yeah. Now, in terms of sort of the current panel that you have, is there any work in progress to expand it further into cancer, into autoimmunity, into other areas, or where do you stand with that?
Yeah, so great question. So we are always working to improve our technology, but the main focus has been sort of to scale kind of clinical validations and scaling up to kind of being able to measure biobanks like the size of UK Biobank and other huge studies with hundreds of thousands of individuals, and making sure that you get consistent results over time and between different spectrometers, and sort of more going to the kind of validity for results back to individuals rather than competing with some of the other exciting technologies where you can measure thousands of metabolites or proteins. Using NMR, we cannot go there. There's kind of, even though we would love to measure whatever inflammatory cytokines or something, there's no way that we can measure that with NMR. So this is one solution that can really be used in large studies, but it doesn't take away the value of doing subsets with deeper omics.
Okay, all right. And I see there's a new question in the chat. Could you speak from Julie? Could you speak to the impact of collection protocols on the assay? For example, when applying a Nightingale biomarker risk score developed in one cohort to participants in other cohorts?
Yeah, excellent question. So we see broadly very good consistency of many of the biomarkers are quite robust to collection protocols. There's some measures like glucose and related biomarkers that are susceptible to how quickly the samples are processed. If they're from the clinical workflow, a few biomarkers may be lost, but broadly the biomarkers are very robust, and many of the studies indeed have been kind of collected in real world clinical workflow. The scientific results with disease prediction are very consistent, and what we have, for instance, biomarker scores developed in the UK Biobank replicate really well in, for instance, the Finnish cohorts, and now we have access to Estonian Biobank at very large scale, and we see, I would say, excellent replication there. In particular, when you look at the same age ranges, that I think there's a more subtle effects as kind of whether new collections have very large fractions on statins or differences in the elderly versus the young are important to take into account, but that is what we now have the sample sizes and different studies to validate.
Yeah. I mean, I think it's fascinating to see sort of, the array of diseases where you showed risk profiling scores. I mean, Jeff, as you know, I mean, for example, in our sort of the pheno codes we have, or ICD-10 pheno codes that are quite well standardized. I mean, there are about 1,600 of them. And, you can run everything against everything, and then obviously also incorporate other omics data into the equation. So, I mean, I think you will see a lot of these type of studies sort of, going forward.
All right, then there's a good question from TM again. Are your biomarkers AIDS and sex specific? So specific is a strong word, but I think they're related certainly to AIDS. Many of them are related to AIDS, but some are following kind of curves like BMI, there where you see increasing values up to AIDS, and then they go down in the elderly, but others are just constantly increasing with higher AIDS. And actually there's quite some elegant work in relation to aging and sort of broad disease risk, mortality risk that one can make really nice biomarker scores for. Then for sex, we often, if you just do kind of adjusting for sex, then it does kind of most of the trick. And we rarely see different association magnitudes for different sexes. But some biomarkers like glycine and glutamine and HDL related markers, the concentration levels are quite different between men and women.
Peter, one other question for you. Have you looked at treatment effects? So effects of diet is an obvious one, but effects of medical treatments, drug treatments in particular, to look at differential response of individuals that may have the same genetic risk, but a different treatment response, which would be important for drug development.
Yeah, excellent point. So the vast majority of studies we profiled are general population, biobanks, single time point, electronic healthcare record follow-up. So I think there's a huge opportunities in trials, in various intervention effects, whether it's lifestyle or pharmacological. There are some legacy trials like some statin trials and diabetes treatment trials on sulfonylurea and other common medications that have been profiled, mainly to understand what are kind of the molecular effects of the drugs. And also a lot of genetic analysis for drug targets that are mimicking new drugs, and kind of some studies that I was involved in showing almost identical results from statin trials and the genetic variants related to statin treatment. But for many other drugs, it's not really well known, like sort of beyond the cardiometabolic space. I think it would be important to study what we see kind of from observational designs, like multiple time points. There doesn't seem to be like tremendous effects of these drugs, but sort of touching upon differential response, we have not yet gotten to that point. The evidence of that aspect of precision medicine is also a good opportunity to move on to.
Thank you.
And maybe a little bit on that sort of same venue, is there any study that has published showing correlation or association between these biomarkers and imaging, brain imaging data, such as MRI data and degenerative changes in Alzheimer's disease? And sort of in that venue, Jeff, I mean, do you think that, let's say you had 500 imaging pictures of Alzheimer's patients with certain changes, but then you would have 100,000 people measured with these biomarkers? Can you sort of do a Mendelian randomization type analysis, not unsimilar to we do with genotypes?
Yeah, have you ever looked at the imaging or in other data sets?
So there are some studies published on this, and there are correlations, but they are so weak that in a way you cannot say the surrogates are so far away from, so you cannot in a way avoid the imaging. But there's some studies, kind of Mendelian randomization studies, in relation to depression that suggest some causal biomarkers in that space. I think those causality studies are somewhat hard to do if it's not that the genetic variants are directly sort of related to the say amino acid transporters and so on. So I think we need multiple lines of evidence to sort of hone in on the causality in relation to neurological diseases, but somewhat promising results.
And when the UK Biobank releases their full imaging data set, which I guess isn't brain, I forget, they have a sort of full trunk one, and then I don't know if they also have a brain scan.
Yeah, I think there's also some brain.
Then I think it'd be a very interesting experiment to do, because it'd be a much bigger sample size, I think, than anyone's looked at before.
Right. Yeah, I mean, we have a very sizable number of just clinical MRIs, in our biobank. So I mean, we would have, dozens of thousands of samples to do that type of analysis. So that would be a future focus for sure. I see there are a couple of more questions in the chat. One is sort of, whether saliva or other specimens, which probably, I mean, I think you're standardized sort of really for plasma
yeah, so we focus almost entirely on plasma or serum samples. We do have a urinary biomarker platform that has really exciting results for kidney disease. I think it would also be fantastic to study in large scale for, say, dietary intake and so on. But it's much more sort of new biomarker discovery, exploratory analysis, being first mover, whereas the Nightingale platform by now is quite established in terms of cardiovascular metabolic epidemiology. I say maybe for other diseases it's more novel, but we cannot measure saliva samples or nasal samples.
All right, I think we are coming close to the timeline here. I don't know if there is any final burning question from anybody, but Latifa sort of, it's basically wrote a couple of comments to thank you all for participating, and particularly you, Peter and Jeff, for your presentation and your time this morning. It was very informative, valuable. I don't know if anyone has any other final final
Yeah, I can maybe
saying?
make a commercial that we kind of have a special offer for IHCC cohort, sort of extending the good work kind of we did with Hakon and others. So feel free to reach out, and we'll see if we can provide support for your research.
Great, excellent. So,
excellent. Thank you so much.
Thanks, everybody.
all right, take care. Thanks again.
Thank you all again, and please let us know if you are interested to present your talk, your research outcome on this platform. Please let us know. Thank you. We encourage you all. Thank you, and happy holidays.
Same here.
Thank you.
Bye-bye. Bye.
Bye.